I want to continue the manufacturing CMC challenge discussion a little bit, which I know we can dedicate an entire hour to just on that topic. But because EVs present unique challenges when it comes to isolation, purification, maintaining product consistency, can you walk us through the current bottlenecks in scaling up EV production, ensuring batch-to-batch consistency, and how are you and your team at Aegle working to overcome any sort of challenge?
So first of all, for consistency, there's not a perfect way to do it for sure, and I'm sure there could be better ways. But first you start with the donor, right? So we isolate our donors, or isolate's not the right word. We confine our donor criteria to a certain age group. Initially, our CSO had wanted to do only male donors, but we couldn't really understand why that would be, valuable. We don't think there's a big difference between those two. So we included female donors, but they have to be, under a certain age. They have to, meet, of course, by the tissue regulations, they have to meet a lot of stringent criteria. So, keeping your donors as consistent as possible is the beginning, right? And then you just keep your manufacturing as consistent as possible. The challenges are obviously that people are different, right? And so one person's good, even if they're the same sort of age, et cetera, they're going to have different numbers of cells get isolated and different productivity of those cells in terms of extracellular vesicles or exosomes. So there always will be that. But I have to say that unless it's a failed batch where the cells die or fail to do well, which is pretty rare if somebody knows, how to do cell manufacture, the range isn't too bad. I mean, it's manageable. And I say that from I'm looking through the lens of someone who's been doing cell and gene therapy my whole life. So my tolerance level for range is way different than someone who's been doing, say, polymer chemistry or small molecule or monoclonal antibodies and has a very defined set of, it has to meet within these tight ranges. At least CBER at the FDA shares my tolerance to some extent, more so than something like CDER or CDRH, the drug and device divisions. So, I think that you kind of do the best you can at this point to keep everything as consistent as possible and recognize that you have to be willing to maybe throw a lot away if it's not meeting what you think it should meet. But you try to keep those ranges as broad as is practical. And one of the things that we'd like to continue to explore is how do those what matters in the ranges, right? So we set these ranges based on what we get, right? That's what everyone has to do when you do this stuff, because you don't know what the right range is. You just know what range you can hit with lot to lot. So then in a perfect world, you go back and you say, well, if it's lower than my lower end or higher than my higher end, does that matter? And so you have to have an appropriate model or set of models in vitro and vivo to check that and say, what difference does it make? And could that range be bigger? We haven't fully, that's a life work, right? So we haven't fully gotten all those answers. But I think that's just continuing to build the field, as a community to know those answers is what's going to have to happen to make it more consistent.
Sure. I'd like to take this opportunity to remind our audience to please submit questions as you think of them. And speaking of, I do think that one came in that we could loop into this conversation now before moving on. And someone thoughtfully asked, do you think automated and closed system processes will be key in the manufacturing processes?
So was that automated? Sorry, what was the question? It's automated and closed system?
Yes, will be key. So perhaps automated or automated or closed system processes. And do you see one or the other making more of a difference?
I don't have a great answer for this. I assume so because, I think in order to try to make things more consistent, that kind of intuitively would make sense. Having said that, having worked in cell therapy, there is an art to it, and we can't instill too much art in GMP manufacture, right? But at least in developing the manufacturing, there is some art in understanding the cells and how they're going to produce what they're producing. So I don't know that it can be fully automated. I think there still has to be some human intervention, and I could be completely wrong about that, but I think that I don't think we're going to fully get rid of human intervention in this kind of manufacturing anytime soon. I think closed system is going to be more critical, just from the fact that you can't really sterilize these products, right? I mean, you can't put them through e-beam or an autoclave or, ethylene oxide. You have to use single-use things. Everything has to be done aseptically. There can be terminal sterile filtration, but you lose a lot of product if you do too much of that kind of filtration and sterilization technique. So I think, closed system is going to have to be really important. Again, I don't know how much automation we can get from that, but yeah, that's my 2 cents.